ये बंद हो गया अभी।
Now let us start.
Okay, sir. Aditi ma'am, shall I start? Assalamu alaikum wa rahmatullahi wa barakatuh. Good evening to all the dignitaries, my colleagues and dear participants. With great joy and immense exultation, I feel privileged to extend my warm welcome to all the participants present on this platform for today's guest lecture arranged by Department of Microbiology, Maulana Azad College, Aurangabad on viral mutation and its consequences. Friends, we all are facing the challenges emerged during the post-COVID-19 pandemic in the beginning of year 2020. An outbreak of a novel coronavirus strain, now known as COVID-19, was identified in Wuhan, China. Its fast worldwide spread led World Health Organization to declare a global pandemic. Since the SARS-CoV-2 variant known as B1617 was first reported in India late last year, it has spread to dozens of other countries, including the United States, Singapore, and United Kingdom. Where it has become dominant in some regions, researchers are now rushing to investigate these variants and work out how they might affect the trajectory of the pandemic in countries where they have gained a foothold. Key questions remain about how quickly the variants can spread, their potential to invade immunity, and whether they cause more severe disease. To answer many more such questions, today's webinar has been arranged. We are fortunate enough to have with us the eminent speaker, Professor Arun Kharat, sir, who will be going to talk on the topic viral mutation and its consequences. Before we'll start with the lecture, I would like to I'm profusely elated to take an opportunity to introduce our chief guest of today's webinar. The whole campus is vibrating and echoing his prominence, the man of distinct vision and fountainhead of illuminating ideas, an idol of knowledge and experience, an inspiration to all of us. Please give warm welcome to the Honorable Principal, Maulana Azad College, Aurangabad, Dr. Mazhar Ahmad Farooqi, sir. I now request Honorable Principal, Dr. Mazhar Ahmad Farooqi, sir, for his keynote address. Over to you, sir.
Assalamu alaikum. Honorable guest speaker of today's program, Dr. Arun Kharat, Professor, School of Life Sciences, JNU, New Delhi, teachers from other colleges, and my dear colleagues and students. We as a college are on the lookout for social issues, fear-mongering, mental health, misconceptions that requires an immediate address. All these and more are the areas that have been identified and addressed by eminent scholars, scientists, philanthropists, and medical professionals through the series of our webinars. They have enabled us to reach out and address our academic and social commitments. In this series, we are delighted to have an eminent speaker, Dr. Arun Kharat, with us today. The SARS-CoV-2 virus has mutated over a period of time, resulting in genetic variation in the population of circulating viral strains. The molecular Antigen and serological tests may be affected by viral mutations differently due to the design differences of each test. The identified potential impact on performance of vaccines in the backdrop of such mutations need to be understood. Let us put to rest some speculations, fear, and unscientific thinking and be enlightened by the expert's opinion today. Dr. Kharat, who has years of experience in teaching as well as research, with him will today deliberate on viral mutations and its consequences in the backdrop of the pandemic. I have my best wishes for you for an enduring scientific journey ahead. Thank you. Thank you very much.
Thank you, Principal Sir, for your words of wisdom.
Thank you.
Thank you, Principal Sir, for your words of wisdom and for your guidance. May I now request Dr. Madhuri Sasrabuddhe Madam, Associate Professor in Microbiology, Maulana Azad College, to introduce guest of honor, Professor Arun Kharat, sir. Over to you, ma'am.
Thank you so much and good evening all. It's a very Herculean task for me to introduce Professor Arun Kharat sir in a very short time, but I will try my best. He completed his post-graduation from Shivaji University, Kolhapur. He did his PhD from prestigious Institute of India, Indian Institute of Science, Bangalore. I'm very happy to report that part of his thesis work has been published in 3 articles. And one of the 3 articles has been referred by Burgess Manual of Systematic Bacteriology, that is Shigella genus. He is a recipient of prestigious awards and fellowships. To name a few, he has received State Bank of India Meritorious Student of Employee Fellowship for high school, pre-university and university level studies. He has received fellowship from Indian Institute of Science by the Department of Biotechnology to conduct research towards PhD. He is a recipient of Nobel laureate James Watson Fellowship for presenting a paper in most renowned international meeting, Genetics of Bacteria and Phages, held at Cold Spring Harbor Laboratory in August 1998. He received fellowship from French Ministry of Science Fellowship to do postdoctoral fellowship with University of Joseph Fourier, Grenoble, France. He is a recipient of Rockefeller Fellowship to do the postdoctoral research in Laboratory of Microbiology, Rockefeller University, New York, USA. He started his career as a scientific assistant C in Tata Institute of Fundamental Research, Mumbai. Because of his passion of teaching and research, he joined as assistant professor in Abasaheb Garware College, Pune. He completed his postdoctoral research from Grenoble, France and Rockefeller University, New York, USA. He worked as a professor in biotechnology in Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. He was a director of UGC Human Resource Development Center of Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. Presently, he is working as a professor in microbiology in School of Life Sciences, Jawaharlal Nehru University, New Delhi. He has more than 35 research articles published in prestigious journals of international repute to his credit. He has published 36 nucleotide sequences in GenBank. Just a minute. Yeah, he has guided students from Portugal, Germany, and USA for their research work. He is a research guide of Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, and 3 students were awarded PhD from the university and 1 from Swami Ramananda Tirth University, Nanded. 7 students are carrying out their research under his able guidance. He is a reviewer of American Society of Microbiology, Elsevier, Springer, Blackwell Publisher, Rockefeller Press, Wiley, Academic Publisher, Science Domain, and Bioinfo Publications. He has worked on various selection committees for recruitment of teachers in various university departments of Aurangabad, Pune, Kolhapur, Udaipur, and Mizoram. He has delivered popular lectures in various colleges and academic occasions, not only in Maharashtra and other parts of India, but many countries such as Sweden, France, Poland, Boston, and Newark. I'm happy and fortunate to have such a multifaceted personality, dedicated researcher, strict administrator, and very good academician with us. I'm proud to report that he is my treasured student. With this brief introduction, I request Professor Arun Kharat sir to deliver a talk on viral mutations and its consequences. Professor Kharat, please.
Thank you very much. Let me take this opportunity to thank Principal Sir for giving me an opportunity to interact with this mob. The Department of Microbiology head, Dr. Aditi Madam, my very own teacher, Madhuri Madam, Nejabin Madam, and a lot of teachers who I see, I know them also. And I like the most part is like a strict administrator. Thank you very much for giving me this honor. So let me share my screen. I made a slight change in the this thing title. Rather than a viral mutation, I have made it like a virus mutation, because the viral mutation could have a different implication, because it could be any other mutation which has become viral. So I think viral mutation perhaps would be an appropriate term. And what are the consequences? And I think Nejabin Madam has already given certain important keyword. Then also Principal Sir, Faruqi Sir also brought an angle to it. But I would go more initially with the academics part of the concept of mutation rate and how this evolves. Then I will land into the second part. I will land into the COVID-19, which is also the acronym given to the SARS-CoV-2. I really like the term also that Indian B617 derivative has spread among the 15 to 20 countries. We have conveniently forgotten that the whole pandemic, the base has been the Wuhan strain, and almost over 170 countries are affected at this moment. And let's go more into it. And I hope that this would be multi-log, so that towards the end, a lot of people may have queries. So basically, there are two terms in mutation related: mutation frequency and mutation rate. So frequency does not include rate, but rate cannot be explained without frequency. Now, basically, it talks about the changes in the DNA or the gene that particular wild type strain converts into mutant, and at what frequency. But this is not a general frequency. This frequency is referred to the mutation probability per cell division, or in a singular organism, or per cell, per gamete, or per round of replication in general for the eukaryotic, multicellular, multi-organelle organism. So it's a very defined and complicated. Now, this is a beautiful illustration and summarizing graphically the whole paradigm or the saga of the whole context of genetic drift, or the rate at which we are amazed to see the variants of COVID-19, which of course I am going to bring a term that what you call amazing is not at all amazing for the scientist, because scientist has seen much wider world, and we have only been focused around one keyword, that is a COVID-19. So everything is in the light of our earlier experience with the diseases, so that the bacterial, fungal, or the viral diseases. And then all of our understanding is being compared, parallel with the COVID-19, and on the basis of chaos, social site, especially India-like country, which is highly dense, and the democratic conditions are such that where, although we have a similar dense country, China, but the way it functions, the administration in China, and the way democracy functions in India is uncomparable. And we could see a huge chaos, and you could see the huge social side disturbance. So, but I think as a scientist, I already have seeded a word that what you are amazed with the changes and with the mutations, and that is very clear from your proposition that came to me a week before, that viral mutation and consequences. That clearly indicated that, oh, people are so much thinking about variants of COVID-19. And perhaps I'm going to give you the surprise that don't get paranoid and don't think that COVID is the rapidest developing strain. So, but anyways, we'll come to that. The graph shows on the x-axis the size of genome and the y-axis the mutation rate. Normally what happens, numerically, the lower side is less and on the y-axis and topper side is high. So 10 to the power minus 11 being the very low and 10 to the power minus 2 is being the very high, the mutation rate. 10 to the power minus 2 means like 1 base out of 100, and 10 to the power minus 9 means 1 base out of 1 billion bases, and 1 base out of 100 billion bases mutation per cell division or per gamete or per cell per replication cycle is calculated mutation rate. So human genome is 3 into 10 to the power 9, while the amphibian genome is in the range of 10 to the power 11 base pairs. The bacterial genomes are in million bases, 10 to the power 6 bases. And the size of genome, if you do compare, of the viruses usually are thousands of bases, like a few 10 kilobases, 20 kilobases, 30 kilobases, particularly COVID-19, the virus in your mind is slightly below 30 kilobase pairs. So it's like 10 to the power 3, we can roughly say. So you can see the viroids are the even smaller genetic material, RNA materials, which are lesser than the 1,000. They encode only for the protein. They don't really have their own machinery. So the viruses, which in themselves are $1 million question as to they are living entity or non-living. So viroid is out of that, meaning that they are very well known to cause a central nervous system diseases, and that also has a genetic material, and that also is subjected for the mutation. So we need to take into consideration of when we talk about a viral mutation, we really have to take into consideration of mutation that can go at a what speed. So you can see the genome size is a small or genome size is a larger. The larger the genome size, you could see the mutation rates are also very low, 10 to the power minus 10 to 10 to the power minus 11 for in general for the eukaryotes, mammals, plants, amphibians. When it comes to bacteria, in general, the mutation rates lies around 10 to the power minus 9. So this is average calculated. Typically, in case of bacteria, 10 to the power minus 8 to 10 to the power minus 10 is a probability of a mutation rate. From gene to gene varies. And so 10 to the power minus 8 minus 10 would be average is minus 9. So it's fine enough. There are also double-stranded viruses. A lot of bacteriophages can be the example of double-stranded virus. There are also animal viruses, plant viruses, which are double-stranded virus, cauliflower mosaic virus, very popular one, and vaccinia virus, pox virus. They are also the double-stranded DNA viruses in the case of animal virus classification. They also have higher mutation rate as to 10 to the power minus 8, 10 times faster than that of the bacteria. But however, their genome size is also 10 times smaller within the 10 to the power 5, 10 to the power 4. So 10 to 100 times smaller genome than that of the bacterial genome. Bacterial genomes are like 100 times smaller than the lower eukaryotes such as yeast and fungi. And they are smaller 10 times to 100 times than the highly evolved plants and other mammals or the amphibian, the largest 10 to the power 11, as I said already, it is not even covered here. Now, if you come to the single-stranded DNA viruses and RNA viruses, whose size usually is 1,000 bases or few thousand bases, their mutation rates are in 10 to the power minus 4, 10 to the power minus 5. It seems to increase. And when it comes to viroids, which are less than 1 kilobase per size or less than 1,000 bases size, and they are RNA sequences, their mutation rate seems to be the very high. That is 1 nucleotide out of every 100 nucleotide. So it is conceivable to say that the smaller the size of genome, the higher the probability of mutation rate. That's a one statement we can try. Then another thing is that if we have to discriminate the nature of genetic material as to the RNA or the DNA, DNA is a little bit subtle, stable to the mutational pressure as compared to RNA. Perhaps this could be an evolutionary region. The RNA was the origin, not the DNA. Still, RNA could not establish as a stable genetic material. Perhaps there was lots of issues with its own mutational possibility. And that's why the highly evolved organisms from bacteria onward, you could see that every bigger organism, genetic material is the DNA, not the RNA. RNA remains a genetic material only in the virus, virion, prions, viroids, etc. Now, the same slide is shown out here. This is taken from a reference, that is why are RNA virus mutation rates are damn high. This has been published in PLOS Biology very recently in 2018. So this shows the depict again the same thing, that the viroids are the most susceptible for the mutation, and viroids are what? Basically, they are again RNA element that causes some troubles to the plant disease and encoding this one. The viruses have examples, Ebola, that also has been very recently caused a pandemic. Single-stranded DNA also has been shown to suffer a little for the mutational pressure. And as it gets the genome to the double-stranded nature of the DNA, so that be a bacteriophage genome or the plant animal virus genome seems to get a little bit stable. So you could see that icosahedron bacteriophage shown is little bit stable than that of the these animal and the plant viruses, as you could see. And the bacteria, again, they have more stability, and the mouse, which has more or less similar size of genome as that of the human in billion bases, it has the mutation rates below 10 to the power minus 10. So it brings a question that whether the mutations are completely at random or mutations are due to some purpose. So let's first take the genome, DNA containing genome and RNA containing genome. What differences do they have specifically? The polymerases. Particularly in the case of RNA viruses, if you have a single stranded, it could be directly serving at itself as a messenger RNA, or it has to be converted back into another form of RNA, that is a complementary RNA. So there it requires the enzymatic machinery to be able to convert from one type of genetic material to the other type of genetic material. So the transcription is very, very important aspect in the central dogma, because transcription converts the RNA genome into the functional RNA. So the difference between the genome RNA and the functional RNA is functional RNA encodes the gene products, whether the primary gene product or the secondary metabolites, but that is the gene product. And therefore, the functional RNA plays a crucial role, whereas the information is being coded into the genome, so that be RNA or the DNA. Now, single-stranded RNA, if that happens to be a negative one, the positive sense RNA has to be made with a complementary, that is RNA-dependent RNA polymerase. But normally, you have seen the replication enzyme in those viruses, RNA viruses, is also RNA-dependent RNA polymerase. Crucially, they have been proven in large number of viruses, they do not really have a mechanism called a proofreading. Rather, they have an editing. Okay, so proofreading plays a crucial role, whether it is a bacterium, whether it is a fungus, whether it is other eukaryotic cell from the plant kingdom or the animal kingdom. The crucial role is basically the proofreading. Proofreading ensures that there is no mutation introduced unless proofread fails. And in the case of double-stranded DNA, you always have one template to identify whether the complementary base is properly added or proofreading needs to remove, and that is called a mismatch repair mechanism. So such kind of repair mechanism preceded by the proofreading are likely to be absent in the smaller sized RNA genome containing viruses, automatically make these genomes vulnerable for the mutational pressure. That's a point number one. And that's a very molecular biology terminology of central dogma. I'm sure most of the people who are layman, non-biological, they will say, "Kya bakwas kar rahe sir? Ye to hamare sir se ja rahe, flight ja rahi hai sir se." But the point here is that without these things, I wouldn't be able to convince you the RNA viruses are more susceptible for the mutational pressure. So mutational pressure doesn't come from external sources. It is an inherent property that is blessing or that is a curse to the genome. Now here you could see that A scenario, B scenario, and C scenario. Now in everywhere you have a 3 coded color: green color, neutral mutations are shown by the orange to a red pink color, and deleterious mutations are shown by the dark red color. Now the scenario A, if you just look at the beneficial mutation, see, mutation does not always mean a bad thing. Sometimes mutation may give you the advantage, and sometimes mutation may not have an advantage. And every mutation may not have anything always to do with. Madhuri madam has taught me this thing in third year BSc 1990-1991. From the steering year, I love this statement that every phenotype has a genotype, but every genotype need not to have phenotype. Means what? कुछ भी बदल दिखता है मतलब म्यूटेशन हो गया, मगर हर म्यूटेशन के बाद बदल दिखना जरूरी नहीं है. तो इसीलिए ना, I'm coming back to the concept, the beneficial mutations which are neutral, meaning they don't contribute in either way, where you can calculate their impact, their whatever, like a good or badness. But there are certain mutations which are deleterious. Now, what do we call a deleterious mutation? Deleterious mutation to virus, not to the host. We are not talking about host. We are talking about a fitness. So take a look at the left side, the fitness. In terms of fitness, some mutations may not make a difference or they don't contribute at all in the fitness. Such mutations are neutral. Some mutations make them better fit in the given situation. So those are the beneficial mutation. And some mutations makes them weaker. Those are the deleterious mutation. Now, unfit. Unfit one is the A and best fit is the B. Now you could see that unfit situation has a high number of beneficial mutation, meaning it has become very comfortable, cozy virus, but then it has fitness being compromised. So what we're talking here, there is a price to pay. So if you have a beneficial mutation, then that would make itself as a weaker in terms of a pathogenicity or weaker in terms of a virus per se. So as you could see that a deleterious mutations seem to be much more in the C variety, whereas the B variety, which is a better fit now, it has already been found. There are no beneficial mutation into the C variety. So ultimately, the genotype at B is more fit than that of the A, and it is closer to a fitness peak. So it has a smaller fraction of a beneficial mutation. So there is a balance of beneficial mutation versus deleterious mutation. Now the C, if you look at, it has highest fitness, but what has happened? The beneficial mutations have been reduced. So virus stability is itself at a stake. And this is a very important point. I would like to bring it to all audience attention, especially when we talk that अरे ये नया वेरिएंट आ गया, ये बहुत इन्फेक्ट करेगा, ये ऐसे करेगा, वो वैसे करेगा, इतनी सारी चर्चाएं चल रही है. But it would, the more it fits into the community, it has lots of beneficial changes. So it itself is subjecting more into the problem. So let me put it into the other words for the COVID. Let's say predator-prey relation, because a host and pathogen. Now, if it is going to kill every human, a day or a point will come when virus would not have a host and it would have to hunt for another host. That is genetic shift, which is not very easy for virus to acquire. And therefore, in a predator-prey relation, both of them in the ecosystem would find an equilibrium, a balance that is again achieved by the genomic exchanges, and a host can't change it. But the parasite has to change it. So the virus would have to acquire the mutations in such a way that it would continue infecting, but it would be less fatal. That is why the B is better fit than the C, if you can consider. So it has to have some amount of, but on the scale, if you see, C seems to be better fit here, like certain variants which are trahi paida kar rahe, wo wale variants. But then they are having a lot of deleterious properties. They don't really have a better fit property. They look like fit right now, but in other scenario, there is no benefit with those mutations and they are going to vanish as compared to the other better fit mutations. So we have to focus on those variants which are going to sustain in the society rather than the variants which are going to create a problem, which are transitory. Now, RNA viruses, perhaps the most intriguing biological entities in which to study the mutation rate. They encode their replication machinery, and that's why they have been optimized to a better fit. And inherently high mutation rates are there with the RNA viruses. They yield offspring which differ at least by 1 or 2 mutations from their parents, producing a mutant cloud of descendants that complicates our conception of a genotype strictness that we already discussed in the previous slide. Their ability to rapidly change their genome underlies their ability to emerge novel host, and this is what has been told in the case of corona virus too, that it was into the other type of a reptile, type of a mammal, and then from that mammal it has made entry into human, and इसीलिए non-veg नहीं खाना, ये बहुत सारी चीजें बताई गई है, but these facts are yet to be confirmed on the scientific ground. They might escape sometime vaccine-induced immunity. This is also the concern raised in the keywords. So of course, these all possibilities are there, but most importantly, because this is an RNA virus, still that situation is not permanent because it will continue evolving, and evolution in these cases are the must. And sometimes they might evolve to circumvent the disease resistance. So the mutation rates have not only been studied for COVID or the influenza group of viruses, but they have been almost 25 different viruses. This is even earlier. The recently one says 25 different viruses, and their mutation rates have been called or studied as a substitution, base substitution per nucleotide per cell infection. So, and that will be correlate to their selection bias, whether it is naturally selected or non-naturally selected using a new statistical method. Now, it has been shown that rates normally ranges between 10 to the power minus 8 to minus 6 for the DNA viruses, whereas it would come to the 10 to the power minus 6 to minus 4, meaning 100 times, at least 100 times more probability for having the mutation per infection, per nucleotide substitution in the case of RNA virus than that of the DNA viruses. So here is again the same situation. It has been shown. This is a summary of a large number of viruses, and it is clear that the probability is too much for the small sized RNA viruses. Now, what are the mechanisms? Basically, because they are highly dependent on their ability to generate a de novo diversity a short period of time. And that is what we have exemplified in less than 1 year, several different variants. And especially we are counting the variations of the mutations only into spike protein of COVID-19. Nobody has been actually talking about a genetic diversity beyond spike protein of COVID-19. So you can see that it is appreciably high. Rates of spontaneous mutations would vary amply among viruses such as RNA viruses. They mutate faster than the DNA. Single-stranded viruses mutate. Then single-stranded DNA viruses are even in the between RNA viruses and the double-stranded DNA virus. Viral mutation rates are modulated at different levels, including polymerase fidelity. So I already mentioned to you about error fixing ability is not there, but every polymerase is born with its own fidelity. So some of the times, large number of viral RNA polymerases are not as proficient as that of the higher level microbes or the eukaryotic systems. They also perform in the context of sequence, the secondary structure adopted by that RNA molecule and the cellular microenvironment where it is replicating. So its a replication quality, its perpetuation quality also depends on the host environment in additional, it means also. And proofreading and access to the replicative repairs are lacking, that is make them more vulnerable, that I already have preluded. Additionally, massive number of mutations can be introduced by some virus encoded diversity generating elements such as a host encoded cytidine and adenine deaminase, because host also has certain enzymes for its own safeguarding mechanism. DAM and DCM. Those are the basically they can deaminate and that deaminated forms of the nitrogenous bases in RNA viruses would subject them for the lethality. So they would acquire the deleterious mutations due to the host cellular deaminases, and that is also going to go against the viruses. So our current knowledge of viral mutation rate indicate that viral genetic diversity is determined by the number of virus dependent and host dependent processes, factors, and therefore the viral mutation rates can be evolved in response to the specific selective pressure. This is not a very old story I'm giving you. This is a very recently published article, Cellular Molecular Life Sciences in 2016. So it's a very recent new knowledge. Now, how the genes are being, genetic diversity is being adopted into the population. And this is a very recent article that I'm bringing in front of you. Population genetic diversity plays a prominent role in viral evolution, particularly its pathogenesis, immune escape, and drug resistance. And these were the keywords that you guys have brought in the beginning to be discussed in the light of COVID-19. So this diversity is subsequently modulated by natural selection and random genetic drift. That means what? Mutation takes place during replication or transcription, but transcription word I have used because if it is a plus to plus strand, so that replication and transcription is same. So in that case, that mutation needs to be fixed, and that is going to be decided by the natural selection pressure where the replication is happening. And what do I mean by where the replication is happening? Meaning the cellular environment also comes into play, as well as the random genetic drift, because that's also a random process of mutation. So there could be basically the Muller's ratchet, I would like to bring it, like irreversible accumulation of deleterious mutations in a population as a result of random drift can happen. And those mutations, they would like to thrive for the equilibrium between the influx of new deleterious mutations in the population and removal of these deleterious mutations through the selection, because mutation might have happened, but the natural selection would choose whether to fix that mutation or whether to eliminate that mutation. If that is going to be fixed, then that is a mutational selection balance. And that is how your B1617, B16172, B1.1.7, P1B 429 have been evolved. These were not the only mutations happened, but these are the mutations which have been selected by the natural selection, and that's why we see them. And what we are talking these days largely in the light of vaccine is that viral causes species, a population of viruses replicating and competing at a high mutation rate. If that happens, then this is something to be a worrisome factor. Now, mutation rate, as I already have talked about this slide, so it could be DNA damage, DNA replication, editing, and also DRGs, which are the factors, and that's why because they don't have 3' exonuclease activity in the case of RNA viruses, they don't have repair possibility, hence they are subjected for high mutation rate. While on the left-hand side, the system has got the exonuclease repair, and that's why their mutation rate is much lower than that of the RNA viruses. Now, again, take a look at molecular determinants which are required for the viral mutation rates. Now, we divide them into the two categories: higher mutation rate giving viruses, other material, genetic material, and the lower mutation rate giving. So global genomic features, normally higher mutations can be seen into the single-stranded short genomes or RNA genomes, while lower mutations in double-stranded DNA or a larger size genomes. Specific template features can be RNA base pairs and sequence repeats, mononucleotide repeat, dinucleotide repeat, trinucleotide repeat. Such cases do not really impact in the case of lower mutation rate. Replication mode normally is a semi-conservative. If there is that is RNA viruses is semi-conservative, then there is a possibility of high mutation rate. But if it is a stamping mechanism, that is basically a complementary, not just a semi-conservative, but also the reverse complementary, then it would ensure that the proofreading will ensure that you have a template which will ensure that the other base added is a wrong base, so let's fix it. Fidelity mechanisms, when they are absent, then the mutation rate, but naturally, because there is no fixing removal of it, and 3' exonuclease and the repair mechanism will reduce the number of mutation or the mutation frequency. And of course, the mutation frequency is a base for the mutation rate, because mutation frequency per cell division, mutation frequency per cycle of replication is what a mutation rate is. Post-encoded proteins, certain times, are there responsible for the, like I say, DAM, DCM, that is the systems. While uracil DNA glycosylase and MUT-H, MUT-L, MUT-As, and DNA adenosine methylation possibility, mixed DNA protected itself from the pressures of mutagenesis. And that's how the mutations are reduced. So systems that safeguard the whole genome is very crucially important for reducing the mutation rate. Other host factors could be unbalanced amount of dNTP pools, because if the cellular system does not have enough of the particular nucleotide and the RNA viruses are replicating, they are going to choose the pool, and that could also result into the imparting certain wrong nitrogenous bases at a wrong position and fix them due to the mutation selection. Then redox oxygen system that could of a cell to cell. And this is a very important point I would like to make a note of it. Now, in the background, initially somebody was playing some post-COVID and particularly mucormycosis. And there was a one keyword of a diabetes used. So what diabetes does? Basically ROS. So the ROS system of the host can favor a viral system, or ROS system of the host can be deleterious for the host itself. ROS can cause chaos to both, confuse to both, or selectively give an advantage to one. Such kind of factors don't really help to reduce the mutation, rather they help to increase the mutations into the RNA type of viruses. So these are the factors that we take into consideration. Now I'm coming to the evolution of COVID virus, which is evolving. Now several new variants have been generated, and they have basically been treated as a new threats. And how worried we should be actually. This has been a very recent article in New Science by the Lawton J. And I would like to say that as early as April 2020, the first variant, that is a P1, was found in Brazil. That was the first variant. And that variant had 3 different substitutions, not together, but 3 different substitutions. That means in 3 different cases, 3 different substitutions of the spike proteins were found. That lysine was replaced with a threonine at 417 position. Then asparagine was replaced with the lysine, which is a completely dissimilar, because asparagine and lysine don't belong to the same class of amino acids, 484 number of amino acid. Then 501 number of amino acid was also mutated, and that was all found into the Brazil. And then 484 particularly was noticed as a first variant. That was the first variant reported in April 2020, which continued then again later on into the January 2021 in Japan. So the same variations were also found. So it's not necessary that it started in India and it is going to be the case. So later on, the UK variant team, which we all know that basically B117, that was 501 as well as 681 positioned amino acid. And this guy, this variant was found to be more invasive. So it was spreading, the spread was more better, that infectivity rate बढ़ गया, मतलब बहुत खराब situation हो गया. No, it's not like that. The question is that whether the variant or that particular pathogen is having high virulence. High virulence means what? That it just does not infect, but it results into the severe clinical outcome, or the clinical outcome can be ensured at the highest possibility, then it is highly virulent. If it is merely infecting without casualty, without much of a fatality, or without much of a complicated clinical manifestation, then such variants may spread in the community very larger in number. However, they don't really take a big toll as far as the casualty fatality is concerned. That was seen with B117 derivative. But the point I'm trying to make it here is that the expected mutation frequency for COVID-19, given that all the factors that I have already mentioned, because COVID-19 has one subunit, remdesivir was targeted against. That subunit has some ability to fix the proofreading. So this is a virus belonging to the RNA group, but has a blessing of proofreading. And due to the presence of proofreading, though it does not have a 3' exonuclease activity, the mutation frequencies of COVID-19 has been significantly lower than that of calculated risk on the basis of its genetic nature and the size of genome. So given this point now, I should be able to convince you that whatever mutations we have seen, we have heard, we have read about COVID-19, they are far lesser than actually, in theoretical sense, it can happen. So this would be a new surprise, a new term for you guys to realize that COVID-19 has a number of derivatives which are not exactly very high. But social psychology, especially the global way they look at India and the Indian psychology, has made it possible multiply or amplify the impact of COVID derivatives more than actually they can do. I'll tell you why and how. Those who really follow closely the news media, the global news media I'm referring, they would agree that during mid of March 2020 or even the mid of April 2020, whole globe was criticizing India, stating that India is not disclosing actual data. India has a lot of cases. India is, then slowly the media started saying India is not testing people, Indian citizens. That's why the number of Indian infection is very low. That means everything was already prejudiced that India would be hitting the highest number of COVID cases in the world. Was it a random? I don't think it was random. Was it then nonsense? No, it was not nonsense. It had two bases. One, sociological basis, perception was there. Of course, that is what you would all agree also. And the most important second basis was the science. The earlier catastrophic pandemic was in 1999 called Spanish flu. And over 1 million deaths globally took place at that time. And now you can imagine a century ago, India's population wasn't 139 crore. So it was much lesser, probably. But still, at that time's global population, India's population was still more or less higher. And out of 1 million of deaths, at least 20% deaths were from only India. So obviously that history was there, that scientific data was there. Spanish flu took 1/5 global toll only from India. And now that India was such a high with the population density, so everybody had to believe, everybody had to think that whatever it gets into Indian administration system, due to Indian constitution and the Indian whatever public administration, it is not comparable to that of China. In China, if you found disobeying, यहाँ डंडे लगाए तो हमने इतने माइंस बना के इतने व्हाट्सएप पे जोक्स करे उस पे, वहां तो शूट एट साइड भी हो सकता है, but nobody can say anything, neither you can make joke about it, but in India you certainly can. So these all points were there. It's a different type of democracy where freedom is slightly more, it's a liberal democracy than that of the more administrative oriented democracy of United States of America. So there is a discipline, there is a responsibility, but there is more freedom that is being used in India. And that is what you can see even in the city when you go, the youth is walking without wearing the mask. So what can we do? But that cannot happen in China-like country. That's why the global prejudices can be some way or other way justified. But then what happened? In the March 2020, it would be additional information to you. India's ability to perform COVID test was 1,600 tests per day. And today, India's testing capacity has crossed 15 to 16 lakh tests per day. So this has improved a lot. And out of all that, if we see the death rate has still been around less than 1.5. So it's not a huge thing given that. But anyways, but the chaos in the numbers, another interesting feature that let's say somewhere in the third week of April or in the beginning of May, the cases were 4 lakh per day. And death used to be 3,600, 3,700, 3,800 per day. Now the cases are less than 1 lakh per day and deaths are around 2200, 2300. So in my calculation, people are happy. Death has come down from 4000 to 2200. So बहुत अच्छा हो गया, सब कम हो गया, सब आराम हो गया. अरे भैया, उस वक्त के म्यूटेशन डेथ रेट के कंपैरिजन में आज का 4 गुना डेथ रेट बढ़ गया है. But we believe in numbers. So this is a clear-cut example that we follow number. We do not follow the typical statistics. You have to calculate the number of infections in the light of population. You have to calculate number of deaths in the light of population, at least 1 million per million. And in that sense, India is not in top 100 country. But still we talk about Indian variants nonetheless. And the globe is also doing it. So the variations have been reported, so many mutational variations, such as a B117, B1351 from South Africa, which came in October 2020, immediately after the UK variant. So the first came in Brazil, then came in UK, then came in South Africa. And after that, I think we hit another variant in Nigeria that was 1525. And then came into India. That is what is a Delta variant. We call it double mutant, triple mutant. So you could see that L452 was already been reported to have the mutation. And it was found to exist in USA also. It was also found to exist in some strains of UK also. It was also found to exist in some of the strains of Brazil also. And P681R was also, P681 amino acid was also found in September 2020 UK. So it is not that these mutations are into totally out of blue, like a new amino acid. They are into the known amino acids. Only the difference is that either the substitution is not same, or there have been strains with 2 different substitutions or 2 different mutated amino acid in a one patient. Or one strain, and that has made it a double mutant and triple mutant possibility. The triple mutant possibility is under question still. Double mutant infectivity has been found, but still the infectivity rate of Indian variant is lower than the Brazilian derivative, and the proof can be still there. Of late, in last week, the deaths among the children from Brazil are increasing, or they are higher. So these are the examples I would like to bring to your mind if you have any questions coming up. Okay. So we already have been through the second wave, and the whole world has also gone through the 2 waves. And do we really expect to have a third wave in India? Yes, because Indian sociology and social system is such that we cannot make everybody see the sense. I'm not talking about a village. I'm not talking about a semi-urban or the urban population. The youth, particularly students from your college, you must have experienced also. They think, "Arey, mujhe kuch nahi hua, main kuch nahi ho raha." But you are carrying the infection to n number of people, your own family, and things can go bad. Normally, the respiratory infections have historically been known to target senior adults and the preschool children, that is under 5-year age. And senior adult was already been invoked from the beginning, Wuhan till date. The reason is explained was ACE2 receptor was unlikely to be there in the infants and the children, and hence they were overruled. But in the second wave, what I know, in Aurangabad itself, few tens of children from few days to 10 years age have been died. So obviously, और मैंने ये भी सुना कि किसी एक हॉस्पिटल का मैं नाम नहीं लूंगा, मैं किसी हॉस्पिटल के स्टाफ ने बताया किसी हमारे दोस्त को, कह रहे वो ACE2 रिसेप्टर बच्चों में नहीं होता था ना, अब बच्चों में वो दिख रहा है, इसीलिए बच्चों में इन्फेक्शन हो रहा है. You think that acquiring ACE2 receptor in the children can happen in night? The child that was born like 8 years ago, it did not have ACE2 receptor. And now that pandemic has started and due to COVID, that child has developed ACE2. No, virus has changed its host binding ability, and it is not into the spike protein, behind the spike protein, which world has not been speaking as of now. But anyways, if all those possibilities do hold in the bounty of the virus, certainly the third wave and especially under 5 can be targeted, no doubt about it. So we need to be mature. जो भी त्रिसूत्री बताई गई है, 2 गज की दूरी, मास्क है जरूरी. Sanitization should be done with the normal soap water, not alcohol-based sanitizer. From the day when I have been against with that, because it can lead to dryness and sometimes skin irritations, and in some cases, possibility of mutation resulting in cancer can't be ruled out at this time point. And third, obviously, a social distancing that we don't want to go out of it. We give an explanation. अरे अभी इजराइल ने नो मास्क थ्योरी निकाली है, यूएस भी जा रहा है. अरे भाई तुम्हारी यहाँ की त्राही सिचुएशन में कहाँ नो मास्क की बात कर रहे हो? But these things needs to be controlled. Otherwise, not third only, we can have many more waves. Do we believe that should we be happy or should we be comfortable that a pediatric braids are made? As far as what my understanding about host pathogen interaction is, that we don't want pediatric braid as a solution, because pediatric braid is not solution. Solution is protecting every child, and that could be, if we can, of course the trials are underway. If our trials comes with the pink flies, then vaccinating as many children as possible would give them a protection rather than creating a bed. Because उसी lecture का background में जो मैंने सुना कि भाई किसी को आपने क्या दिया, डेक्सामेथासोन की बात कर रहे थे, हाँ स्टेरॉइड दे और ज्यादा स्टेरॉइड देने की वजह से ये होगा. अरे अपने यहाँ पे कौन सा डॉक्टर क्या कर रहे हैं उस पर किसी का कोई कंट्रोल नहीं होता है भारत में, राइट? So it is not like America that if you give 1 ml extra, doctor का certificate निकाल देंगे, because doctor can't give 1 ml extra because he doesn't get it freely. यहाँ तो अपनी दुकान, अपना ही फार्मेसी, अपना ही सब कुछ होता है, मन में आए लगाए, मन में आए निकाल दे. So should we be at a peace? I don't think based on the pediatric braid we should be at peace. No, we can't be. We can be at peace provided that vaccination trial comes with a success and a positive outcome, and then we actually go conduct vaccination of the children as much as possible. Particularly under 5 children don't really have high amount of antibody capacity, developing capacity, and that's why they need to be more protected. So vaccine and COVID-19. इसमें एक बात तो जरूर है कि कोविड-19 की वैक्सीन के जो जवाब है लाजवाब है अभी तक के, मतलब सारी इम्यूनोलॉजी की थ्योरीज जो भी है पूरी उल्टी सीधी करके रख दी है. So some people say that the antibodies can be detected after 1 year. Some people say that after 3 months antibodies are completely absent. So we cannot make any general rule in the case of COVID. It all depends on the individual, whether it was a mild infection, asymptomatic infection, severe infection, whether it was active recovery or the passive recovery. The more active recovery and no comorbidity is likely to have a longer sustaining immune response as compared to somebody with comorbidity or passive recovery. Passive recovery means आपने स्टेरॉइड लिया है, ये लगाया वो लगाया, मतलब आपने बॉडी से रिकवर नहीं हुए. What should be the gap between the 2 doses? लोग न्यूज़पेपर में पढ़ते हैं, फिर से गवर्नमेंट बदलती है. Of course बदलेगी. The reason is that Covishield का data that we always used is a UK data, and whatever the immunological protection efficacy varies, and accordingly the gap between the Covishield particularly will change. Now we have a Sputnik, now we are expecting to get the Pfizer and large number of vaccines. But a rule of thumb is that vaccine is vaccine. It's meant to protect you. So there is no need to worry about a vaccine and its efficacy. You can choose and just go for vaccination. Should recovered people take 2 doses? That's also a question. If you are comorbid, probably tomorrow policy may come that they need to take 2 doses. At this moment, 1 dose is sufficient for a person who has recovered. Current policy says after 3 months, which is acceptable. But if somebody has a multiple comorbidity, probably a single dose is not going to be helpful in that patient. Are we protected? To a great extent, after vaccinations, we are protected, even if one contracts a disease or the infection, because disease is a result of virulence. Infection is just a test positivity. It is not a virulence. So the quality of infection, the quality of clinical outcome will be definitely much milder than the quality of outcome in that individual had him or her not been vaccinated. So certainly the vaccination is going to protect individuals. So should we go for each year vaccination? This question is a very coming up question. फिर लोग बोलते हैं क्या अगले साल हमें खुद लेना पड़ेगा? अरे भैया वही तो होता है ना, शादी में जब जाओगे तो खाना एक बार मिलता है, दूसरे टाइम का खाना तो घर में मिलेगा. अभी तो कोविड का जो चल रहा है उसमें गवर्नमेंट स्टेट हो या सेंट्रल हो कर देगी, बट एवरी टाइम यस इन्फ्लुएंजा वायरस और कोरोना का पहला जो आया था 2002-03 में, उसका भी एवरी स्प्रिंग वन नीड टू टेक अ वैक्सीन बिकॉज due to the mutational genetic drifts, there is a lot of variations possible because these are the small genomes RNA viruses. So new variant may not be sufficient enough to give the full protection. I'm referring to full protection. It does not mean that that individual won't have a protection at all. And as I said, some individuals have antibody titer after 1 year. Some individuals don't have antibody after 3 months. So we cannot make a general statement whether everybody should have a vaccination every year, but theoretically, one should have it. Post-COVID, particularly the health implications are a big thing. COVID has told us very loudly, crystal clear. If people haven't heard, then they are at loss. Maintain your health. So work on your health rather than work on paying money, getting bed, getting oxygen, getting steroid. These all amenities can be purchased, but health cannot be purchased. Health has to be earned. So spend 2 hours, 1 hour every day for your own health and develop yourself from within so that you are completely prepared, not just to take up the COVID, but any such kind of infectious pandemic or infectious epidemic, you would be able to fight with without getting into the complications. So certainly you're going to be prepared, avoiding any kind of communicable diseases, obviously. Thank you very much. I'll be happy to take questions if there are.
Hello.
Yes.
Yeah, good evening. So I will request Arif sir to unmute Rafiq Sheikh. You want to ask question? Arif sir, please unmute Rafiq Sheikh. Rafiq Sheikh, please ask question.
He has to unmute from his side. Message is sent to him.
Please unmute Rafiq Sheikh.
Yes, I have sent the message.
Okay.
He is not unmuting from his side.
Okay, shall I read out the question?
Hi sir, Rafiq here.
Yeah.
Actually I have posted that question in group. So my question was like, can mutation in the coronavirus make vaccines completely ineffective, or it doesn't matter? Like if we have some 1 or 2 doses, and then in case mutation happens, so will that impact vaccination?
A type of vaccine and the quality of health of that individual is of paramount important factors, rather than generalizing a term, whether it will completely eliminate the protection or there will be a partial protection. So this would completely depend not just on the mutation that has been taken or newly added into the virus, but how fit that individual is, and what is the immunological antibody sustainability maintained by that individual. That is a very important aspect. So of course the viruses are going to be variants are going to be produced, and before also all influenza viruses, including SARS-CoV-1, original one, was also having a numerous derivatives. Even now also H1N3 has been generated last month. So it keeps on changing. So there is no guarantee that mutation won't take place, but it is not necessary that that mutant derivative will completely eliminate, particularly Covaxin like virus, where the whole virus is there. So it's a polyclonal response, right? So if the mutation takes place in one particular antigenic determinant, possibility is that other antigenic determinants are protected. So in that individual, the kind of sufferedness you would observe would be much weaker than the kind of sufferedness without vaccination. But again, how much of a gap is between that vaccinated time to the new virus? And in that, if that person has a comorbidity, person has some sort of weak immunity, then of course that candidate may have completely eliminated the immune response already.
The next question is from Afreen. Afreen, please ask question.
I think she is saying, will there be more variations in third wave, and will third wave be deadliest wave?
Yeah.
See, variations are taking place even before the second wave. We have witnessed the infection had come down in October, September, October. That was also not because of environment. It was because the virus was continuously mutating. It was getting a beneficial mutation, but the beneficial mutations will make it less fatal, right? So obviously, that's why virus was eliminating, and this is a process of virus-host equilibrium. So in that case, of course, a mutation is a continuous thing to happen. We can expect to have the variants, and as a human being, we don't talk about the variants which have weakness, meaning the weaker virulence. We are only bothered about the variants, those have been highly infective or highly virulent. So possibility is an open possibility. During third wave, or third wave will be the result of such variant, cannot be explained at this standpoint. It could be a deadliest strain has been created, and that results into third wave. It is not that third wave will create a new variant. There's a possibility that variant is generated and that results into the third wave.
Yeah, Namira Mariam. Namira Mariam.
What is her question? I think.
Madam, ask them to unmute first.
Yeah, please unmute yourself, Namira Mariam.
If she is around, I think.
Yeah, Namira, please unmute and ask the question. Aishwarya is there on the line? Aishwarya Sobnath.
I think she had asked the same question. Namira Miriam had asked the questions, but other perspective, that can mutant be produced with a reduced V pathogenicity? I think I have answered that. September, October time, all the variants were of the reduced pathogenicity, and they were even less infective also. But then the February 2021 variant was the more spreading, and I think it wasn't as deadly as the original one was, but still people believe it. And this can be used for vaccine production. Now there is a concept of mixed vaccine. So I think the mixed vaccine would be better than using the weakened viruses. Idea बुरा नहीं है कि जो derivative weak हुआ है उसी को use करो. But again, it is a possibility that the same vaccine, if it goes into the individual, might turn out to be vicious for that particular individual. You have to consider and respect the human diversity also. So that cannot be done. Reduced virulence by means of a laboratory is a feature. That certainly you know that it will not be a virulent in the human, but the strain that has been reduced, its pathogenicity naturally cannot be directly used as a vaccine. It should not be, because in one incidence it might be a weak, but in other individual it may not be a weak. So that cannot be used as a vaccine.
There is another question from Afreen.
Herd immunity is out of box.
Afreen, you unmute and ask question. Hello.
She's asking the
Hello,
herd.
sir.
Yes, Afreen, go ahead.
Yeah, Afreen, please ask question.
Good evening, sir. Thank you very much for okay, I understand about herd immunity, but I also wanted to ask that there have been production of oral vaccines which use different mechanism of action. I would like to ask, will mRNA targeting vaccines are much more effective, or will oral vaccines will be more effective?
See, oral is a route of administration. It is not a nature of vaccine. See, what you are talking, two things you are talking. You are talking about the oral vaccine. That just tells you the route of administration, whether it is a spray nasal vaccine, whether it is orally consumed vaccine, or whether it is parenteral route, that is by the injection into the blood or IM. That is what you are talking. That you don't mix it with the mRNA vaccine, because mRNA is a type of vaccine. So type of vaccine and a route of administrating vaccine cannot be compared at a time. You know what? But oral vaccine, the polio has been used, but the global scientific community has always contraindicated. It should not be given oral. India was the only oral prescribing. IPV, injectable polio vaccine, has been globally accepted. India used to do once and they stopped later. So oral consumption is not a bad idea, but absorption of the oral vaccine to the target site is basically a big question. So it would be not the same amount of the vaccine shot as that of the IV or IM or the nasals, to some extent even the nasal. But oral will be a weaker vaccine, or they have to provide a more dense material as a vaccine in that case. The mRNA vaccine works well as of now, not a problem. It has been.
There is one more question from Aishwarya Ballavde. Aishwarya, please unmute yourself and ask question. Aishwarya. Mohammed Saqib is there? Mohammed Saqib, please ask question. So I will read out the question of Mohammed Saqib. He has asked that after COVID-19 patients, how it causes mucormycosis.
It's a very simple answer. Mucormycosis does not occur in every patient. Okay. It occurs only a very small fraction. Actually, even the severe COVID, that is cytokine storm, takes place in around less than 1% of infected population. And out of that, less than 1%, that means how much? It's a 0.001% of the actual infection subjects for the mucormycosis. Another feature which is in tight connection with mucormycosis is a diabetes. So if the person already has a diabetes, borderline or identified case, then consumption, your treatment of dexamethasone or prednisolone would reduce the immune function and at the same time increase the sugar level, which makes it favorable for the fungi. And if the same patient was placed on or treated with the oxygen therapy, so obviously the humidity is also in the favor. So all these factors together makes to perpetuate fungi more easily into the respiratory path, and it spreads all the way up to the eyes and the central nervous system. So diabetes, then steroids and the oxygen therapy are the factors that are to be considered for the mucormycosis in COVID-19. Mucormycosis is not a new disease. It had been there. It is just that it was only subjected for the diabetic patient or the immunocompromised. But treatment of COVID-19 imparts these two conditions more at severe level, and that's why the mucormycosis you could see in them. Okay.
Yeah. Namira Mariam, please ask question. Namira, unmute yourself and please ask question.
Good evening. I have a question related to the patients who have recovered from the COVID disease. There are some cases have been come forward about people suffering from heart diseases, heart problems related to heart. So is it due to the remdesivir, the steroid, or is it due to the mutated viral strain of SARS-CoV-2?
It's due to the cytokine storm of the viral infection. So let's not attribute that to a mutation in the viral strain. That's the one thing. Second thing is that if those patients who already had a comorbidity, particularly diabetes or the coronary vascular disorders, such as a blood pressure or triglyceridemia or the cholesterol disorders, or they have already had a myocardial, some kind of risk factors, all those patients are more subjected for the thrombosis. And patients who have a thrombosis risk factor, they certainly could have a heart attack. It is regardless of their age or the physical outlook of that person, it could have. It is not necessarily due to a particular strain of a virus. It is the internal health. That's what I'm saying. The health of the individual is more important than the mutational nature of the virus. So the more healthier you are, the more chance that you are going to fight back the virus. The more weaker you are, the more comorbidity you have, the more chance that you are going to suffer with such kind of infections. Thank you.
Yeah, thank you so much. I hand over to Major Bin now.
Yes, ma'am. Thank you. Thank you, Professor Arun Kharat, sir, for enlightening us on virus mutation with respect to mechanism of virus mutation, Muller's ratchet, significance of proofreading, and about different strains of COVID-19 and waves, significance of social distancing and sanitizer, vaccine, etc. And also for answering all the questions in such a lucid way. You have made this difficult topic very easy to understand for all of us. Once again, a big thanks to you, sir. And thank you, Dr. Madhuri Sasrabuddhe, madam, for introducing the guest of honor and conducting the question answer session. One announcement, all the participants are requested to fill a feedback form. The link has been shared in the chat box. I may now request Professor Dr. Aditi Bhattacharya, madam, Head Department of Microbiology and IQAC Coordinator, Maulana Azad College, for vote of thanks. Over to you, madam. Aditi Madam.
Am I?
You're not audible, madam. Madam, you're not audible.
Yes, yes. I just lost the connectivity, I suppose.
Yeah, please.
Am I audible? Am I audible now? Okay. Thank you
Yes,
so
yes.
much.
One request is please
Yeah.
share the feedback link again. The participants are asking
Okay.
for the feedback link. Please.
It is given. It is given. It has been given. The feedback link has been given in the chat box.
Okay. Thank you. Thank you so much.
Hello. Yeah.
Yeah.
Yes. Yeah. Honorable Principal Sir, respected Dr. Arun Kharat, my colleagues and dear students. I first of all offer my heartfelt tributes to our late mentor, Padma Shri Fatma Rafiq Zakaria ma'am. We also share our grief with all families who have lost their near and dear ones to this deadly virus or people who have suffered this virus. The pandemic for me has been a teacher, having taught several things over and over again in the past 2 years. Diligence, cautiousness. It has asked us to be a learner and to be aware of several things and above all, be very, very patient. I start with the thanks that I have to offer to several people here. My first thanks to Principal Sir for planning this series of webinars as part of our social as well as our academic commitments and for giving us this opportunity to be a part of it. My sincere thanks to Professor Arun Kharat sir for his extremely lucid lecture that has taken the understanding of viral mutations to a different level. I am very sure that the students will be taking back a lot to think and ponder. From the routine discussions that we have been having on webinars with respect to COVID-19, that mostly hovered around the spike proteins and probably the ACE2 receptors. This is definitely a new dimension. The discussions on RNA polymerases, the RNA genomes, and the helplessness of probably the RNA polymerase for not being able to correct the strands that it is synthesizing. So it is definitely a new dimension and students should take back a lot from it and prepare their project work or research accordingly. I'm also very sincerely thankful to all the team members of this webinar, particularly Dr. Arif Pathan sir, in particular, for being so gracious and so patient with all of us. He has been managing these webinars, he has been hosting, sitting through all these webinars, and he has been doing so graciously. So thank you so much, Arif Pathan sir, once again. And to all my team members, the staff of the department, Dr. Madhuri Sasrabudhe and Ms. Majabin Saeed, for always being there and to take up any kind of challenge. I am also extremely thankful to all the participants who have spared that time to be a part of this wonderful lecture. Our alumni, Rafiq Sheikh was just asking a question, and there are several other alumni and several other college teachers who have joined from other colleges too. Thank you so much. And that is all from my side. Please attempt the feedback link so that you can give your feedback and also get the certificates. Thank you once again.
Thank you very much. Thank you.
Thank you so much for a wonderful lecture.
Thank you, ma'am, for having me.